I know, right? I know.
Okay, there's the cell cycle done. It's not really a cycle. And the reason, it's kind of funny, it's referred to as the cell cycle, but it's really not a cycle because when you go through it, you don't come out at the end the way you were at the beginning. You got two cells. So it's really a one-shot deal. But because it is cyclical and it's generally unidirectional, it's easier to just kind of think of it as a cycle. So what we're looking at up here, this is just the generic, very, very generic, stereotypical cell cycle for a typical eukaryotic, particularly a mammalian cell. Okay? So the duration is about 24 hours. Although you're going to see in a minute, we can shrink it down to 10 minutes if we have to. So it's about a day. And if you take a look at a cell that just finished dividing up here at the very top, okay? Cell completed dividing, went through mitosis, and we have two daughter cells, okay? Those cells typically do not continue dividing. But instead what happens is most of these cells, after they finish being born, they actually exit the cell cycle. And they go into this little state called G0. And G0 is kind of like a holding area. And it's referred to as quiescence. And in that case, what happens is, and this is actually where you hope, most of your cells at any moment in time, that's where they're at. Okay? They're metabolically active. They're capable of macromolecular synthesis. They're doing normal stuff. But if you want to get them to resume entry into the cell cycle and get to where they can replicate their DNA and go through another round of cell division, you have to treat them with an extracellular growth factor. This is kind of setting the stage for the RTK signaling we'll be moving into. And that is the function of extracellular growth factors, which are also referred to as mitogens, because they promote mitosis, is that what they do is they're going to go through a signaling pathway that's going to cause cells to exit G0 and to re-enter the cell cycle. And then the cell can go through that process one more time, go through mitosis, and you have to do this every single time that cells divide. Cancer eliminates that step right there. So cancer isn't about cells necessarily dividing faster. What you typically do is that you lose the requirement for an extracellular growth factor. So whereas normal cells can check out and go into G0 and hang out there indefinitely, cancer cells lose the ability to do that. And the reason is that generally there's going to be components that work in these RTK signaling pathways that have undergone mutations and have become oncogenes. And they're transducing a pro-proliferative signal even in the absence of an extracellular growth factor. Okay? G0 is nice because it's the only place normal somatic cells can check out of the cell cycle and stay there. As you're going to see, if we try to do that at any other point in the cell cycle, the cells can do it transiently, but then they die. They're going to commit apoptosis. And that's one of the protection mechanisms cells have to avoid accumulating mutations that could, in fact, result in cancer. Okay? So let's say we've got cells arrested in G0. We've hit them with a mitogen. They re-enter the cell cycle. And we'll get into all the details on that in another couple of weeks. They go through a growth phase called G1. It's about 9 hours. And during G1, what the cell is doing is getting poised to replicate its DNA. It's going to cross a restriction point. And the restriction point is that now it's committed to going through a round of DNA replication. And now we're into S phase. And you can see that takes a long time. During S phase, the cell needs to make sure it replicates every single nucleotide in its genome once and only once. That's the key. If that doesn't happen, the cell will stop in S phase and not allow it to go any further until the damage or replication is completed. Failure to do that, the cell exits the cell cycle, goes into apoptosis, kills itself. Okay? So, then we get to G2. G2 is setting us up for mitosis. The main agenda of G2 is to first get everything wired up and ready to go for mitosis, to change some events that occur in the nucleus with the chromosomes, and then out in the cytoplasm with the cytoskeleton. Those are going to be the main components. Mitosis is so complicated, even though it's the shortest point of the cell cycle, we're kind of taking care of that in its own little separate mini unit. It's going to be part of the cell cycle, but we're going to focus on that process all on its own. The main purpose of G2, in addition to getting poised to enter mitosis, is to make sure the cell doubles in size. That's the key. It's a real growth phase. And so the idea is that now the cell will double in size so that when it divides and completes mitosis, which you can see is pretty short, we wind up with 2 daughter cells that are the exact same size as the original cell. Okay? So in general, what we're going to be looking at is using these different systems, Saccharomyces cerevisiae, amphibian oocytes and eggs, and the fission yeast, Saccharomyces pombe. We're going to be looking at different aspects of the cell cycle. Saccharomyces cerevisiae, the budding yeast, we're going to focus on this transition up here. When we look at amphibian eggs, we're going to be focusing on this part of the cell cycle. And then the fission yeast is going to tie in and kind of amplify that. And in essence, what we're doing is looking at how do we regulate a group of serine-threonine kinases that are known as cyclin-dependent kinases. How do we regulate those in order to drive this particular cell cycle? Okay, now, here's an exception to the rule. And that is we do have other cells that can, some of us have them, cells that can exit the cell cycle and remain arrested almost indefinitely, decades even. And these are immature eggs or oocytes, okay? you take a look at them experimentally, these are Xenopus oocytes up here. The cells are really nice and big. They're about 1 millimeter in diameter. You can get thousands and thousands of them out of a frog. Easy to manipulate, very large. And what they do is that they are arrested in G2 of meiotic prophase 1. That means they've already replicated their DNA. Same is also true, but on a much, much smaller scale with human oocytes. You can see the difference there in size, 1 millimeter, 50 microns. They are also arrested in G2 of meiotic prophase. And in fact, human females have all of the oocytes they're ever going to have before they are born. And they're just hanging out in G2 of meiotic prophase 1 for as long as you're around, okay? But what happens is that during ovulation, when these oocytes are exposed to the hormone progesterone, they have to complete meiosis, and then they're poised and ready to be fertilized, okay? And the problem is there's a lot of movement. We basically have to take chromosomes and segregate them in order to complete meiosis. You basically go from a cell that's tetraploid to haploid. And the problem is that that process, as oocytes get older and older and older, the chromosomes don't segregate as well. And so that's why there's a heightened risk of certain types of chromosomal abnormalities if women put off having children to a later age simply because of chromosome missegregation during meiosis. For example, you can wind up not getting a separation of the 2 copies of chromosome 21. They move together. And that can result in individuals that are trisomic. One chromosome 21 comes from a father, but 2 that came from the mother, so they're trisomic, and that's the cause of Down syndrome. So the point is, this process really isn't designed to be on hold for decades, okay? But that's where the oocytes hold up. So oocytes are unique compared to somatic cells, because somatic cells can arrest in G0 indefinitely. Oocytes can arrest in G2. Just a different mechanism of doing it. Now, remember that I said a typical mammalian cell cycle is about 24 hours, okay? We can speed that up. And the way that we can speed that up is that if you take a look at what happens with amphibians, like Xenopus up here, take those eggs, oocytes, complete meiosis, and now fertilize them, and this is what you see here. Okay, so that's a fertilized Xenopus egg that is undergoing its first 12 cell divisions. It's from a frog. It has as much DNA as you and I do. So Xenopus has a genome of about 3 billion base pairs, just like us. Takes us about 9 hours to replicate our DNA, and all that other time, about 24 hours to undergo cell division. This guy's doing it every 10 minutes, okay? They're dividing as rapidly as bacteria. So all these cells are doing after fertilization is that they replicate their DNA. They still have to do that. And they undergo mitosis. So every time you see the embryo divide, it's completing a mitosis. And it's going 1 cell, 2 cell, 4 cell, 8 cell. It does that 12 times. So it does it for the first 12 cell divisions. And what you wind up with at the end is a little embryo that's got 4,096 cells. You can count them if you want to. It's occurring very rapidly. And the reason it is because that egg, that big honking egg that's about 1 millimeter in diameter, stockpiles all of the proteins and other molecules that our cells need to make every time they go through G1 and G2. So the amphibian egg comes up with a strategy to where it can make those first cell cycles go very rapidly by pre-storing everything in the egg that you would need for G1 before you replicate your DNA, and then during G2. But you notice what happens here. The cells are dividing without getting bigger. This is what you're going to see. This is a perfect example of the Wee phenotype. These are cells that are getting smaller and smaller and smaller every time they divide. But they still replicated their DNA entirely, once and only once. So what you're doing is you're taking this egg, and every cell originally with the mass of the egg has a nucleus. What happens is that every time those cells divide, you still replicate your DNA, you still have a nucleus, but the cells are shrinking in their size. So at the end, each one of those individual cells at the blastomer stage is 1/4096th the mass of the original egg. Okay? So the point is, under certain circumstances, we can dramatically speed up and accelerate the rate of cell division if you have to. Now that is not what cancer cells do. Cancer cells don't do this, they just keep dividing, but they still double in size before they do so. So this is a unique situation here. You can also notice that thing's bouncing around a lot as it divides. And that's because there's a lot of movement that's going on among those cells as they go through these very, very early cell division cycles. Okay? Now, there's a price you pay for this. And that is all the cells are dividing synchronously. If one of them has a problem, or has an error in DNA replication, the checkpoints that will allow that cell to stop dividing, fix the damage, and then catch up with its neighbors doesn't work. So the price you pay for that rapid cell division is what you're going to see is that DNA damage checkpoints don't work. They're not there. So what does that mean? It means that for a typical embryo, when you come out at the end of this with 4,096 cells, anywhere from 1% to 5% of them are defective and damaged, and they die. They commit apoptosis. And guess what? You still get a frog out of it. So not all of these cells are essential to going through the rest of development. Okay? But the take-home is you can take your time to divide slowly. And we have all these checks and safeguards that are built into the cell cycle that allow us to make sure everything happens correctly and when it is supposed to occur. You speed that up like you do here, you lose the opportunity to fix things. All you can do is get rid of the defective cells at the end after they've already completed those first 12 cell divisions. So it's just kind of an interesting strategy. All right. Here's what I mean about these checkpoints. Okay? The cell cycle itself is going to be driven by these characters here that are known as cyclin-dependent kinases, or CDKs. They are heterodimeric serine-threonine kinases. The cyclin subunit, as you're going to see, comes and goes. And so what you're going to see is that what's going to cause the cells to advance through the cell cycle is we're going to see different cohorts of cyclin subunits appear on the scene, and then they're going to be degraded. So they have to show up, hook up with a CDK, the catalytic subunit, and they're going to be phosphorylating different substrates. And the CDKs that are during G1 and S are going to be required to get you into DNA replication. The cyclin CDKs that appear during G2 get you into mitosis. They're serine-threonine kinases, so all they're doing is phosphorylating things. The kinase is only active when the cyclin subunits are there. So that's why they have the cyclin-dependent name attached to them. The other way we're going to regulate them, though, is that we can regulate it by phosphorylation and dephosphorylation of the catalytic subunit itself. So having a cyclin hook up with a CDK isn't the entire story. We're going to be regulating the activity of the catalytic subunit, and this is where tyrosine phosphorylation is first going to appear. Everything we've had up to this point, serine-threonine. Tyrosine phosphorylation is now going to play a major role in controlling the regulation of the cyclin CDKs. And where these is going to become important is during that part of the cell cycle there, going from G2 to M to make sure the cell doubles in size before it proceeds into mitosis. Okay? These are the checkpoints over here. The targets of checkpoints are the CDKs. The CDKs are driving the cell cycle. If you want to stop or pause a cell at any point in the cell cycle because something didn't happen correctly or something was damaged, and there's two ways we can look at that. You can damage your DNA, or you can fail to replicate it completely. That means the cell has a sensor that's monitoring the integrity of DNA. And that sensor is going to activate a DNA damage checkpoint that is going to inhibit the cyclin CDK and stop the cell cycle. The other thing that we can do is that during mitosis, we have to form a mitotic spindle in which all of our chromosomes are attached to the spindle so they can be segregated. Our cells have a mitotic spindle checkpoint that's looking at the spindle and says, is everybody attached? Is everybody good to go? And the cell will not go into anaphase and move the chromosomes apart until that happens. Okay? These checkpoints hold the cell cycle up. And the reason they're doing it is they're going to allow the cell to activate enzymes that can repair damaged DNA. So the first response of the checkpoints is to try and fix it. The protein that's going to do that, that underlies the DNA damage checkpoints, as just a heads up, is the p53 tumor suppressor. That's the one protein that underlies virtually all of this. If you have deficiencies in p53, your checkpoints are not going to work. So, what's going to happen is you activate a checkpoint, you stop the cell cycle. You don't want to go any further. Cool. Now we try to fix the damage. You fix the damage, the sensor says, yep, damage is done. The cell will pick up and resume the cell cycle. That happens most of the time. But if you don't fix the damage, then the checkpoint remains active, and now the cell is going to trigger apoptosis. And what happens at that point, the cell is committed to suicide. In multicellular organisms, our cells would rather undergo apoptosis and do themselves in than run the risk of proceeding into mitosis with chromosomal damage or DNA damage. It's a way of ensuring the genome integrity, okay? So the checkpoints are going to be working by affecting the activity of the CDKs, all right? Okay. Organism number one, the budding yeast Saccharomyces cerevisiae. We're going to use a genetic screen to identify genes that are involved in the cell division cycle. So these genes are called CDC, cell division cycle genes. This is what cell division in the budding yeast looks like. Now here's what's interesting. This cell division is asymmetric. So there's a mother, there's a daughter. This mother is going to divide before this daughter did, and there she goes making a new daughter. You can see that the new cell emerges by budding, so that's why it's called the budding yeast. It's also called brewer's yeast. This is the major yeast that's used in fermentation. We can use the formation of that bud to see where we are during the cell cycle in Saccharomyces cerevisiae. So if we take a look at this, down here, when we go from G1 to S, as soon as we start DNA replication, that's when the bud, it's trying, come on, right there. That's at the beginning of S phase. As you continue through the cell cycle, you guys can see the bud gets bigger and bigger and bigger, okay, until it finally separates and buds off altogether. Here's the phenotype that Hartwell used to come up with his mutant screen. So what Hartwell is going to do is to generate temperature-sensitive mutants in yeast. Okay? We've been there and done that with the SEC pathway. Now what you do is you got all these mutant yeast, and what you do is you shift them all to the non-permissive temperature. Well, what's going to happen is that if you affected a cell division cycle gene, the cells are going to arrest at a particular point in the cell cycle. They're going to arrest where the function of that gene is now required. You can tell where they're at in the cell cycle based on, do they have a bud? If there's no bud, it means they arrested here. If you have a little tiny bud, they arrested somewhere right in here, going into S phase. If they have a bigger bud, then it means they arrested out here in G2 to M. So that's how phenotypically Hartwell was able to do this mutant screen to identify CDC genes. Okay? So he did this, and he discovered more than about 100 genes. So these are all different yeast genes that if you ship the cell to non-permissive temperature, some of them would have little tiny buds here, so they arrested in S phase. So that means you compromise the function of a gene that encodes a protein to go into DNA replication. Others had a little bit bigger, so these were genes that were required to go into G2. Others made it almost to the very end, so that means that they were arrested in mitosis. So you go through and you categorize them all. You give them names just like you did sect genes. CDC1, CDC2, CDC3, CDC all the way up to 100. Most of the genes that he discovered with this screen gave little tiny or no buds. So what that meant was these are genes that are required to go from G1 to S phase. So the majority of the CDC mutants that Hartwell discovered with his mutant screen were genes that are required for cells to begin DNA replication, to move into S phase. So it's just an example of one of the genes that he discovered. It was gene number 28, so it's called CDC28. And CDC28, as you see, it encodes the catalytic unit of a cyclin-dependent kinase. Okay? So imagine now, this is back in the early 80s. You've got Hartwell's group that's going through cataloging and identifying all these genes in yeast that are required for cell division cycle. Cool with that. All right? Put that on hold. Now we're going to look at a different system with a different group of people that are looking at a biochemical assay to ask how do you go from G2 to M. Oh, before we do that, forgot. Remember CDC 42? Well, you got till Wednesday to remember it, okay? CDC 42, remember we have RAC, Rho, and CDC 42. Well, now you know what a CDC mutant is. So CDC 42 was the 42nd CDC gene identified in yeast. Okay, guys, why would CDC 42 be discovered as a CDC gene? There's something going on in this video that should tell you where you need CDC 42. Right there. What's happening to the plasma membrane right there? There you go. It's involved in bud formation. That's why CDC 42 was discovered as a CDC mutant. It's basically involved in specifying where do you form the bud and starting to push out the plasma membrane just like you would if it was a cell that was undergoing migration. So there, now you know the answer. Where does CDC 42 get its name? Okay? All right. Now we'll switch gears. So Hartwell's making CDC mutants. Now we're switching over to amphibians, sea urchins, aquatic organisms, and we're going to be taking a look, how do we find the cyclin subunit? Okay, because one of Hartwell's first genes that he discovered was a catalytic subunit. Well, where did they find the cyclin subunit? This is work that was being done in Tim Hunt's lab, so we'll call it the hunt for cyclin. He's working with these oocytes, okay? So here's how he's using a biochemical approach. We can't use genetics here, sorry. If we take an oocyte, it's arrested in G2. We add the hormone progesterone, and what it does, it completes meiosis, pops out a little polar body there, and now it's ready to be fertilized. But what it is, it's arrested in metaphase of meiosis 2. Forget about meiosis, Okay? Here's what I want you to think about. The cell starts off in G2, it arrests in metaphase M. G2 to M. Whatever is necessary for the cell to go from G2 to M gets switched on in response to progesterone. Cool with that. Progesterone is triggering somehow the activation or the synthesis of something that's needed for a cell to go from G2 to M. If you treat the cell with an inhibitor of protein synthesis, cyclohexamine, and treat it with progesterone, it won't go from G2 to M. So progesterone is triggering the synthesis of a new protein that wasn't there before that's necessary to drive the oocyte from G2 to M. Here's how Hunt proved that something was there, that there's a new protein. Hunt figured, okay, if there's something new in this cell that wasn't there before, I ought to be able to take an extract, take that egg, smush it up, make an extract, and inject it into the immature oocyte. So what he's going to do is transfer cytoplasm from this cell that's been treated with progesterone and inject it into an oocyte that's arrested in G2. If there's something new that was made that wasn't there before, then whatever that something is ought to allow this cell to also go into metaphase and arrest. Well, it worked. So he called this activity, we don't know what it was at the time, he didn't know what it was. He called it maturation promoting factor, MPF. The reason is it promotes maturation. This whole process here of going from G2 to M is called oocyte maturation. He took it a step further. He then wanted to know, well, is there something like this in any cell that's undergoing mitosis that you could find? And the answer was yes. So he went to yeast and made an extract from yeast cells that were in mitosis. He injected those into the oocyte. They went into meiosis too. He looked at HeLa cells. He looked at any cell he could find. And as long as you made the extract from a cell that was in mitosis, that activity was present. It was never found in a cell during interphase. So because of that, he kind of broadened the definition of MPF. It isn't just called maturation promoting factor, it's also called mitosis meiosis promoting factor. But it's still MPF. So what Hunt started to do then was to look and see, can I find evidence biochemically of a protein whose appearance coincides with the appearance of MPF activity? I mean, this is suggesting there's something there, but can you find evidence for it? So, this turns into a pretty cool story. Hunt would always go to Woods Hole at the marine biology lab every summer. And he was always involved in teaching a physiology course at Woods Hole. It's a summer course that undergraduates could take. You guys could take it. He decided to have this undergraduate course try and find MPF. So what they're working at, okay, so Woods Hole's up there off the coast of New England, it's off the coast of Boston. Lots of sea urchins up there. So now they're using sea urchin eggs. And so what he had the students do was to basically take sea urchin eggs, fertilize them, and now what you notice is that the sea urchins go through these same sort of cell divisions, very, very similar to what the Xenopus embryo did. 1 cell, 2 cell, 4 cell, 8 cell, 16, so forth. Here's what he noticed then, is that if you take those embryos after fertilization, so here's where they start going through the first cell division, 1 cell to 2 cells. Then it comes down. It takes about another 60 minutes for them to go into the second mitosis. And so what Hunt does then is he says, okay guys, I want you to look at a protein gel. And can you see any proteins that are starting to come up that would correspond with the cells going into mitosis. He found one. So this is now a protein gel, where you're looking at the proteins that are being made at different time points after fertilization. Now, if you take a look, most of the proteins here, you don't see any change in their appearance or disappearance. They're just kind of hanging out there all the time. Not a big deal. We found one protein up here, though. And this protein, you notice it starts off at a very low level. And you notice its activity comes up, and it gets more and more intense. And then right before the cell goes into mitosis, it disappears. Its level crashes. It's being degraded. So the protein comes up, gone. Now if you wait, you notice the protein comes back up again. And then it crashes again. The protein, because of this periodicity in its appearance and disappearance, he called it cycling. So that's how cyclins got their name, okay? Is that these are proteins whose appearance and disappearance tracks with the gain of a cell going into mitosis or not, okay? Now, you notice that throughout the course so far, we've shown pictures of scientists of what they looked like when they got their Nobel Prize. But you probably didn't see what they looked like when they did the work that got them the prize. So it turns out that if you go to the MBL archives and their website, they take group photos of the people that were in the class at the time. There's the banner, Physiology, 1982. Okay? So this was a group picture of the physiology course in the summer of 1982. At the same time, these guys discovered cycling. Okay? So you can kind of date yourself back about 30 years. Here's Tim Hunt. You know, he's kind of a typical Englishman, you know, kind of got the rugby socks going and everything. So you can, first off, you can see people didn't dress very formally back then. Everybody was kind of loose and everything. And that's because Woods Hole's down on a beach. You're always down playing beach volleyball, sunning yourselves, playing around, and then collecting massive numbers of clam eggs. Well, in that summer, I was doing a postdoc in Baltimore at the Carnegie Institution's embryology department. And so I was doing work on Xenopus genes, and so Tim invited me to come up to Woods Hole and give a seminar. So, yeah, I went up. That's me. And anyway, I just happened to be there. That's what yeah, it's summer, dude. That's what we wore. So I show up, and I give a seminar on the work I was doing in the postdoc lab and everything. And we had just been out playing volleyball before then, so you can see nobody was dressed up. And the photographer showed up, and I said, I'm not in the class, Tim. I don't, oh, no, get in here. So anyway, I stood up there in the back. So Hunt goes, okay. So Tim says, you know, you got to clean up your act a bit. So he said, you know, because I told him, I said, well, after this meeting, I'm going down from Woods Hole to Cold Spring Harbor. So the Cold Spring Harbor Laboratories is on Long Island, as many of you know. It's a very prestigious center for DNA research back at the time. James Watson was the director, okay? So after going to Boston and going to Woods Hole, I was going to go down to Cold Spring Harbor and give a seminar on my work there at one of the symposium. And Tim said, dude, you've got to really clean up your act. This isn't going to do it. So he said, you know, kind of look a little sharper, a little more professional, okay? So I took his advice. All right, here goes. Yeah. There it is. The years of teaching this course have not been kind. Man. But look, I cleaned up, you know, I shaved, actually put on a clean shirt. I actually wore long pants, like a belt. You know, that just reeks of 1980. Anyway, so that was the picture they took there. So I was giving a talk on the work I'd done on my postdoc. And you can see I already had the style down. Notice there's no PowerPoint. Look what I'm having to show everything on. You know, I was like, that's all we had back then. This big honking microphone and everything. It sort of looked like I'm giving a blessing. And no clue what I was saying. But there's one thing I want to point out that you guys need to see. This is why this was really cool back then. See that chalkboard? See it? It meant something to me. It's the same chalkboard that Jim Watson, when he first presented the double helical structure of DNA back in 1953, that's the same chalkboard. Same chalkboard. I got to write on it. So like this was like a pilgrimage, to basically, this was holy ground. I'm giving a seminar at the same place, and you can see Watson wasn't dressed up any better than I was. In fact, he was younger then than I was here. So Watson is here, he's wearing short sleeve out. And anyway, that's the first public presentation of the double helical structure of DNA. And so then here I am, 30 years later, and I get to use the same chalkboard. So the chalkboard's gone now. It's been replaced with whiteboard to show PowerPoint and everything on. So anyway, this is what people look like when they don't win a Nobel Prize. This is what they look like when they do win one. And that's also what they look like when they win one. So the humbling side of all of that is to understand that when scientists are really doing important work, or not, they don't look like the dignified dudes in the tuxes that are meeting the King of Sweden and getting their prize and their big cash money. But one thing to make a note of, Cold Spring Harbor still teaches these courses every summer. And they are incredibly important. They're very useful. They are highly informative. And if any of you have aspirations of winning a Nobel Prize, something to notice is that going back to 1920, 54 people that have taken these courses, either as faculty or students, have gone on to win a Nobel Prize. So if you want to heighten the odds, take one of the courses. But I actually put the link for their REU program up here. It's also in Colab. So if you're interested in looking at taking one of these courses during the summer and you want to go up the Woods Hole, do it. It's really cool. It's fun. Okay, meanwhile, back at the cycling. Get rid of that. Okay. So we're still following cycling and MPF. So cycling comes and goes. MPF comes and goes. And here's where Hunt takes it to the next level. So you can sort of see how this story develops. He finds a protein that you would predict is some part of MPF because its periodicity matches MPF. And here's where he really shows it. So several years later now, after purifying MPF activity from thousands and thousands and thousands of frog eggs, they identify the protein. They clone the gene, express it, they make antibodies. Back then, that took a long time. You can see this paper was published in 1990. They published the cycling paper in 1983. It took a long time back then to clone a gene, purify a protein, make an antibody, do all that stuff. Companies didn't exist to do that back then. So it took a very, very long time. So as it turns out, Hunt was actually then purifying and cloning the cyclin genes from Xenopus. Okay? So here's what you find. If you look up here at this graph on the top, you're tracking MPF activity. So MPF is absent in the G2 arrested oocyte. You add progesterone, comes up, we go into meiosis 1, we go back out, then we go into meiosis 2, and we stay there until fertilization. Then MPF activity comes down, and then it goes up periodically every time for those first 12 cell divisions. So MPF activity always comes up right before the cell goes into mitosis, and then it crashes, and it comes up again. If you do a Western blot now, it turns out Xenopus has several isoforms of cyclin. The first ones that they discovered, they designated as cyclins A and B. And what you can see if you do a Western blot, take a look. Here's the appearance of cyclin B after fertilization. The protein's there, so that corresponds right there. It drops. We go into the first cleavage division, comes back up, drops, comes back up again, comes back up again, comes back up again. the magnitude or the amplitude of the change is different from one cyclin isoform to another. But here's the key. As MPF activity comes up, so does the cyclin level. As MPF activity drops, cyclin drops. So cyclin activity, cyclin protein level tracks perfectly with MPF activity. So this is what was really started to nail the connection between MPF as an activity and cyclin as being an important component. Now at this point, you guys notice they don't know what it does. It's just there. It could be all of MPF. It could be part of MPF. You don't really know exactly what it's doing, but you need it, and it correlates with MPF activity. So here's what we find now. So fast forward to the present. It turns out there are 4 main types of cyclins. The first ones that were discovered were the cyclins that are involved in going from G2 to M. They're called mitotic cyclins. They were the first ones discovered. They didn't know there were going to be others. They didn't know how many others. So these guys are called cyclins A and B. Okay? So anytime you see cyclin A and B, you're talking about a G2 to M cyclin. It then turned out there was another group of cyclins that were discovered afterwards. And these were cyclins that go up and come down in order for cells to go into G1 and to go into S phase. So they were discovered after cyclins A and B, so they're called D and E. There is no cyclin C. I'm not sure how it got skipped. So we've got cyclins A and B, and we got D's and E's. D's and E's, as you're going to see, function to go from G0 to G1 to S. D's and E's function before A's and B's. A's and B's are necessary to go into mitosis. D's and E's are necessary to go into DNA replication. Okay? They are the regulatory subunits. They are not the catalytic subunits. The catalytic subunits are over here, and they're called the CDKs. And it turns out we also change the CDKs during the cell cycle. We got CDK 1, 2, 4, and 6. I don't know what happened to 3 and 5. Don't worry about the numbers of the CDKs, okay? We're not going to get into the distinctions of the numbers. What I want you to know and just focus on is that you need a cyclin D or E CDK to go into S phase. You need a cyclin A or B CDK to get into mitosis, okay? The cyclin subunits, as you're going to see, are regulated by selective degradation. And in fact, what you're going to see is cyclins specify their own destruction. They're self-destructive proteins. They're suicidal. And Hunt, as you're going to see when we come back on Monday, Hunt was the one who discovered sequences within the cyclins that are called destruction boxes. These are amino acid sequences that say, take me out. Okay? So what we need is, you need the appearance of a cyclin CDK to move into the next phase of the cell cycle. You degrade the cyclin subunit, that turns the kinase off, and now you can move out. Okay? All right, so that's where we'll pick it up on Monday then. You get your first batch of cyclin questions set, okay? Have a good weekend.
Yeah.
I have fair one.